Hello, everyone. Good morning, good afternoon, good evening, wherever you are. Welcome back to the second edition of Fundamentals of Solid Formulation webinar series. This is a 7-week webinar series by a leading expert who understands your daily workflow and will share his or her real-world experience in supporting preclinical and clinical projects. And we have an excellent lineup of speakers in the 7-week webinar series, starting with Henry Morrison from Gilead, followed by Sudhakar Garad from Novartis. And last week you heard Luca Isolino from Merck. And today we are having Daniel Joseph Price from Millipore Sigma. And entering into May, we'll have 3 webinars, starting with Luca Russo from GSK, followed by Alish Medek from Vertex, and Adam Keats from Syngenta. So this 7-week webinar series is organized by XLPi and JSTAR Research. XLPi is an algorithm-driven artificial intelligence-based pharmaceutical technology company with sites in Boston, Beijing, Shenzhen, and Shanghai. JSTAR is an innovative CRO based in New Jersey and is an independently managed subsidiary of Photon. Photon is a contract development and manufacturing organization based in China. The 3 organizations are strategic collaboration partners in pharmaceutical drug design and preclinical projects. My name is Sivakumar Segran. I'm the Senior Director of Business Development at XLPi in Boston. I'm going to be your moderator today for this FSF 2021 webinar series. Before we move on to today's webinar, I'd just like to give you a brief recap of what happened in the last 3 weeks. So the talks from Henry Morrison, Dr. Sudhakar Garad, and Dr. Luca Isolino are all captured and it's recorded. It's on the website. So if you go to xlpi.com and if you click on the events page and you go to FSF 2021, you can see the recordings available there. So you just have to click on the play button and then you have to complete this form that you see below here. Please fill this form to view FSF 2021 webinar videos. So you need to fill out that form and then you can watch the recordings along with the Q&A page that is also available on the website, where questions that were not answered during the live session are answered by the speakers and then posted along with the recordings. So I'd like to encourage the audience today to use the questions tab to send in your questions during the live session. Please don't wait for the presentation to end because questions will be taken on a first come, first served basis. So also put in the slide numbers that you're referring to, then it allows the speaker today to take in a few additional questions during the live Q&A session. So today we have Dr. Daniel Joseph Price from Millipore Sigma. Daniel is a strategic marketing manager at Millipore Sigma in Germany, and he handles the portfolio of solubility enhancement and sustained release excipients. Daniel plays a key role in several key innovation initiatives, including 3D tablet printing and computational formulation development. And Daniel's expertise lies in mesoporous silica and thermodynamics of amorphous formulations. So today he's going to talk about amorphous formulations for solubility enhancement, risk, and opportunities. So without further ado, let me hand over control to our speaker today, Dr. Daniel Joseph Price from Millipore Sigma.
Good evening, everybody from Germany. Good morning or good afternoon, depending on where you're located across the globe. Big thanks, Shiva, for the introduction and also the opportunity to share with you this presentation today. And thank you all for signing up and showing your interest in this very exciting topic. So today I'm going to talk to you about amorphous formulation for bioavailability enhancement, and specifically look at some of the risks associated with this formulation choice and highlight to you some of the opportunities that are currently available to counteract these risks. We've already heard from Sudhakar in this webinar series how important solid form is for oral bioavailability, and I'd like to build on that today with some specific examples. So what will we cover in this webinar today? Firstly, I will reintroduce amorphous formulations. What are they? Why do we need them? And how can we make them? And then I'll really spend some time focusing on a relatively new type of amorphous formulation technology, which is mesoporous silica. We'll then revisit some of the basics by talking about amorphous stability. Why is it important? How can we predict it? And what do we do when we don't have good amorphous stability? And then finally, wrap up with a summary of everything that we discuss. Okay, great. So amorphous formulation. So I think some of this will be a reintroduction of what Sudhakar shared a couple of weeks ago, but it is important to set the scene, and so I hope I can do that in the following slides. As we know, poorly soluble drugs are on the increase. It used to be that we could take for granted that our drug in our development pipeline would have very nice physicochemical properties, and as such will have total and complete bioavailability from our gastrointestinal tract, and we won't even have to worry about it. But what you can see here is the situation is not so clear today. We are getting a rise of the so-called poor soluble drugs, so those are BCS class 2 or 4, and even more when you look in the future. So in this example, 60% of the NME development candidates in the pipeline were poorly soluble, but some reports in the literature say that that number can be as high as 90%. So if 90% of our small molecule candidates in our development pipeline are poorly soluble, this is a big risk for the overall longevity and success rate of our pipeline, because those poorly soluble molecules, when they're delivered orally in a tablet or a capsule, they must be in solution in order to pass from the intestinal fluids into systemic circulation. So if our molecule is not in solution, then we can have low and variable absorption from the GI tract, lowering the overall efficacy of that dosage form, with a lot of the dosage form simply passing into the waste and being unable to have a therapeutic effect. So we must do something about these molecules. We cannot just put them in standard tablets and just accept that most of the molecule will not be absorbed, because then we cannot have a therapeutic effect. So there are various ways that we can alter or enhance the solubility of these poorly soluble molecules. Medicinal chemists can alter the molecular structure, include some more polar groups perhaps, maybe a prodrug or a salt. But more often than not, the chemical structure of the molecule is strongly related to the efficacy, so the binding of that molecule to its biological target, and therefore the options for altering the synthetic chemistry of the molecule can be limited. So instead, more often than not, we turn to formulation. And one of the ways in which formulation can be used is to alter the solid state of the molecule. So specifically, the solubility can be enhanced using the amorphous form. So what is the amorphous form and why can it enhance solubility? Most molecules that we find in development are naturally occurring in the crystalline solid state. From an energetic thermodynamic perspective, oftentimes the crystalline solid state is a thermodynamic equilibrium. That means it's a state of low energy, and therefore the molecule is relatively comfortable in that environment. The problem is, in a crystalline solid, we have a crystal lattice that holds our molecules together. So these crystalline interactions can be quite difficult to break. And we must break those interactions if we're able to solubilize that molecule. First, we need to remove an individual solute molecule from the crystal lattice, and then and only then can we insert that molecule into a cavity in our solution, thus solubilizing the molecule. This comes with two key energy requirements. Firstly, we need to have enough energy in our system to break the crystalline interactions that hold those crystal lattices together. This is associated with the crystal lattice energy. Then we need enough energy to create a cavity in solution and insert that individual solute molecule into that cavity. This is associated with the solvation energy. So the solubility of a crystalline solid is a function of the crystal lattice energy and the solvation energy. There's not much we can do about the solvation energy. This is typically associated with the polarity of the solvent and the polarity of the molecule. But what we can do is reduce substantially the amount of energy required to break those associations. We can use a different solid state, the amorphous solid state, chemically identical, but no longer held together in a crystal lattice. And therefore, if you simply balance thermodynamics on each side of that equation, energetically speaking, you require less energy to break those interactions apart, and therefore solubility of the amorphous solid becomes substantially higher as it's simply thermodynamically more favorable. The one caveat is that the amorphous solid is not a thermodynamic equilibrium for most molecules. The amorphous solid has an excess amount of free energy, and the system does not like this. It wants to get rid of that excess energy, and it does this via forming crystal lattices. So our amorphous solid would typically recrystallize. So in order to take advantage of the potentially enhanced solubility that the amorphous solid form can provide us, we need to stabilize this high energy, more soluble solid state. So the most common way in which we can stabilize this more soluble solid state is via polymeric amorphous solid dispersions. At a fundamental level, polymeric amorphous solid dispersions are simply a mixture of a drug and a polymer. There are two main ways in which we can process this mixture of drug and polymer. The first is using a solvent-based approach. We can dissolve the drug and polymer in a common solvent. We can then spray dry this solution, and we get what is known as a spray dry dispersion or an SDD. Alternatively, we can take advantage of heat via hot melt extrusion. So we mix, melt, and extrude, and what we get at the end is known as a hot melt extrusion formulation or a HME. Now, the only difference here is the processing method. On a molecular level, these formulations are basically identical. In this formulation, the polymer acts as a matrix and immobilizes the mobility of the drug. So what do I mean by that? Basically, our polymer acts as one big mesh, and you can kind of think about this like a fishing net. In a fishing net made of nylon, our polymer replaces the nylon, and the molecule is in the individual gaps within that mesh, in that matrix, and this prevents those molecules from coming together and recrystallizing. So it stabilizes the amorphous solid state. These technologies have now been used for decades and are also included in commercially available medications, and so they have a lot of credibility and history. But new technologies are emerging. And one of the new technologies to stabilize the amorphous solid state is mesoporous silica. So now I'd like to tell you a little bit about mesoporous silica. Mesoporous silica is simply silicon dioxide. And on this slide, we have the example of Partiq SLC. Chemically speaking, silicon dioxide is relatively simple and has been used for decades in oral solid dosage forms, primarily as a glidant. But with mesoporous silica, we are not just talking about regular old silicon dioxide. Mesoporous silica, as the name implies, is porous in nature. So you can think about mesoporous silica like a giant molecular sponge. And because of that high porosity, our mesoporous silica has a very high surface area. So approximately 500 meters squared per gram. So this is an incredibly high surface area. To put this into perspective, if you were to have a vial with 10 grams of this product, you would have the same accessible surface area in that vial as a standard Premier League football pitch. So a huge amount of accessible surface area. The other important property for mesoporous silica is the pore size. Mesoporous, by definition, is anywhere between 2 and 50 nanometers in diameter, so a relatively large range. But for this mesoporous silica, Partiq SLC, the pore size is approximately 6 nanometers in diameter. So it's from these two very important properties, surface area and pore size, that the functionality of this excipient arises. Specifically, mesoporous silica stabilizes the amorphous form via adsorption to the porous network, so taking advantage of that high surface area. And then secondly, via nano confinement, so taking advantage of that 6 nanometer pore size. So how does it work in practice? We take our poorly soluble crystalline API. We can use organic solvents to break that crystal lattice for us, so to do the hard work of breaking those bonds. We solubilize in an organic solvent such as acetone, ethanol, methanol. This concentrated solution of our API can then be combined with mesoporous silica, and exactly like a molecular sponge, that concentrated API solution is absorbed inside that mesoporous network. We can then remove the solvent rapidly, and then what we are left with is the individual drug molecules confined within that porous network. And because we're talking about a 6 nanometer pore, what we essentially have is a sterically stabilized amorphous formulation. And some really interesting physical chemistry takes place on this scale, and we're going to cover that in the next section. But we have a sterically stabilized amorphous formulation. This powder can then be combined with a standard blend and tableted or filled into a capsule. This dosage form is then taken by the patient, and what happens is basically the reverse of the loading process. So the powder hits the gastrointestinal tract. Like a molecular sponge, that silica absorbs the GI fluids from the gastrointestinal tract. This generates a concentration gradient, so you've got lots of drug on the inside of the silica and no drug on the outside of the silica. And so naturally, by a simple diffusion process, the drug is rapidly released into solution. And because we have the amorphous form, we generate supersaturation. So supersaturation is concentrations higher than the thermodynamic solubility. And this can ultimately increase the absorption across the gastrointestinal tract and improve the bioavailability of the molecule. The one caveat to this whole process is that as soon as you release that supersaturated API into solution, that is an unstable energetic environment. Your molecule does not want to be supersaturated. The molecule wants to precipitate out of solution and recrystallize into that thermodynamic equilibrium of the crystalline solid in solution. The silica at this point has no impact whatsoever on maintaining the supersaturation of that system. And so therefore, an additional component is required, known as a precipitation inhibitor, usually a polymer, which maintains that supersaturation so that absorption can take place over physiologically relevant timescales. So this entire model that I've just described has been reported and described as the spring and parachute model. The spring in this case is our mesoporous silica that generates that supersaturation, and the precipitation inhibitor is our parachute that prevents that rapid reprecipitation. So that, in a nutshell, is how mesoporous silica works, via adsorption to the pore and nano confinement. So let me share with you a case study on this technology, a very basic case study. Well, firstly, how do we actually load these molecules? It's a very important point. The good news is that loading onto mesoporous silica is feasible from lab to production scale. At a very basic level, mesoporous silica can be loaded using simply a rotary evaporator. And we have now validated this technology at commercial scale manufacturing, so up to 150 kilogram loadings. The exact process behind this scale up is a little bit out of scope for this current discussion, but if you do have an interest in this process, then please feel free to get in touch and we can discuss it in more detail. So here's the case study. We took fenofibrates, which I'm sure you're all aware is a standard BCS class 2 compound that is regularly used in the scientific literature. We dissolved fenofibrates in acetone, and we used a wetness impregnation method to load fenofibrates onto silica. And if you'd like to learn more about what that is, again, please get in touch. And as you can see from the right-hand side with our differential scanning calorimetry curves, we have crystalline fenofibrate in black with its characteristic melting endotherm, the downward spike. And then when we have fenofibrate loaded onto mesoporous silica in blue, we no longer observe that characteristic melting endotherm, which means that we have successfully stabilized fenofibrate in the amorphous form. Another important point is the residual solvent. We have dried, in this case, to well below the residual solvent limit of 0.5%. And this is a really important process control step to make sure that the solvent is well below the ICH limit. And this is something that you can pretty much do with no problem from this mesoporous silica. So what is the impact of this formulation on the overall performance of this BCS class 2 molecule? In order to assess that, we carried out an in vitro biorelevant dissolution and an in vivo pharmacokinetic study in pigs. We had two different formulations for the mesoporous silica. We had a capsule, so it was simply filled into a capsule, along with HPMCAS, which was a precipitation inhibitor. In addition, we had a suspension, which is typically used in preclinical trials, and we combined our fenofibrate-loaded silica with the polymer in a suspension. We then compared both of these dosage forms to crystalline fenofibrate, so unmodified fenofibrate, that was also blended with the same polymer to rule out any co-solvency effects. So what did we see from the data? On the left-hand side, we have our biorelevant in vitro dissolution. This was conducted in FASIV. The suspension is in blue, the capsule is in pink, and the crystalline fenofibrate is in purple. And what you can see is that both the suspension and the capsule significantly outperformed the crystalline fenofibrate. And if we look to the right-hand side of the screen to our PK curve, you can see that both of these formulations significantly outperformed the crystalline molecule. In green and in purple, we have the AUC, the curves, the exposure curves of both of these formulations. And you can see that the bioavailability of fenofibrate in this instance was much higher than the crystalline fenofibrate. So as said, this was a relatively simple and straightforward overview into this technology. I just wanted to really exemplify some of the points that I raised in the previous section. This technology is now routinely being used for ECS class 2 compounds. But the more interesting aspect for mesoporous silica is how it has a part to play in stabilizing molecules that are typically very difficult to formulate in the amorphous form. So let's revisit this slide. As we discussed, polymeric-based solid dispersions are the most common formulations to stabilize the amorphous form. On a molecular level, these formulations are a mixture of drug and polymer, and in an ideal scenario, we would have what is known as a solid solution. So this is shown by the figure on the right-hand side. In a solid solution, the drug and the polymer are homogeneously distributed. That means you have a perfect mixture of that drug in that polymer, and as a result, a solid solution is formed. The solid solution is the ideal scenario for a polymeric amorphous solid dispersion, because if you ensure that your drug is fully homogeneously combined, then you have a bigger stabilization effect of the polymer on that amorphous API. The problem we have is that polymers are not so, let's say, inert. They can move about, they have mobility of their own, they can react to changes in temperature and water. And actually the molecule can move about within that polymeric matrix, so it's not totally immobilized in space. And so what we can have is a so-called heterogeneous system or a phase-separated system. In this system, we still have an amorphous formulation, but what we find is so-called drug-rich domains. So that means within that polymeric matrix, we have areas that have more drug than polymer. And vice versa, we have areas that have polymer-rich domains, so more polymer than drug. So that means that the system is no longer a solid solution, it's no longer homogeneous. And if we have a molecule that is not very comfortable in the amorphous form, you've just provided them with the opportunity to recrystallize, because that's what they really want to do. And so what we can have in those circumstances is a totally phase-separated formulation in which our amorphous API has been able to separate out into drug-rich domains and then recrystallize, forming a crystalline phase within the polymer. In this example, we may see a slight increase in the dissolution rate, but we will not see the amorphous solubility advantage. So we will no longer generate that supersaturation. So this is a big issue, also from a long-term stability perspective. If we have an amorphous formulation on the market, we have to be absolutely sure that we will not see such phase separation and recrystallization across the shelf life of the product. Because if a patient receives this middle scenario with a homogeneous distribution, they're getting a totally different dose than a patient who receives a phase separated or even recrystallized system. So this is an important topic. So this is something that I studied in detail. And the question that faces formulation scientists is, how can we de-risk this entire process? How can we make sure that our compound will not phase separate and recrystallize in our polymer. And one of the ways in which we can do this is by determining the glass forming ability of our molecule. The glass forming ability is essentially the propensity of a molecule to recrystallize. Avramov in 2003 described it as the ease of a vitrification of a liquid upon cooling. Essentially, if you have a liquid and then you solidify that liquid back to a solid, how likely is it that that liquid will recrystallize? Based on this theory, Baird et al. Developed a framework to experimentally determine the glass forming ability of a molecule. In short, they proposed DSC as a method to determine this, and they placed the molecules in a DSC pan. They melted the molecule to remove the crystallinity. They then cooled it right back down to solid and then melted it again. And they observed the differential scanning calorimetry curves in this melt-cool-melt cycle. And depending on if and when they observed crystalline behavior, they categorized molecules into 3 different buckets. So firstly, we have the GFA class 3. These are also known as good glass formers. These are molecules in which no recrystallization was observed in that entire melt-cool-melt cycle. These molecules are very comfortable in the amorphous form and have a low risk of recrystallization. In the middle, we have our GFA class 2, our moderate glass formers. These molecules remain amorphous in the first cycle, so they can be melted and cooled and not recrystallize. But as soon as you start to melt that system again and supply a little bit more energy, then they recrystallize, so you observe a melting endotherm. So these molecules have a moderate stability in the amorphous form, a moderate propensity to recrystallize. And then finally, we have the so-called poor glass former molecules, or GFA class 1. These are molecules that when you melt them down to remove the crystallinity, as soon as you start to solidify, as soon as you start to cool down, that molecule will crystallize. This is sometimes known as cold crystallization. And that means that even as you are proactively reducing the energy in the system, there is still an energetic driver to recrystallize. That means that these molecules are not stable in the amorphous form. They have an incredibly high propensity to recrystallize, and as a result, can be really challenging to formulate with polymeric amorphous solid dispersions. It's no surprise then, if we look at the data below from Wittenbach and Kuntz, that the distribution of commercially available amorphous solid dispersions is weighted heavily towards good glass formers. Simply put, good glass formers are way more likely to be successful in an amorphous formulation than a poor glass former. But can we really throw these molecules away and discard them only because of a glass forming ability problem? Or can we turn to different formulation technologies to successfully formulate these potentially high value compounds? So I think it's no surprise, based on the storyline so far, that I would like to share with you some data describing how mesoporous silicate is a really great option for these molecules. Can we take these so-called pore glass formers and leverage this very, very small pore, this stabilization on the nanoscale, to stabilize these molecules where previously they would not? So before we really look into that case study, I'd like to think about the physical chemistry of what happens in this process. What happens when you take a molecule and you put it inside a 6 nanometer pore? And some of our colleagues in Lisbon did exactly this, and they took the molecule menthol. Menthol is an extremely poor glass former. This molecule is not stable whatsoever in the amorphous form. To put that into context, menthol has a glass transition temperature of around -60 degrees Celsius. So there's a standard rule of thumb that those of you working on this might know, that if you have a molecule with a glass transition temperature, you need to store that formulation at 50 degrees lower than the glass transition temperature, just to be totally certain that your formulation will not recrystallize. So we've all seen in the past year just how difficult a cold transport chain can be with the COVID-19 vaccines of -90 and -70 degrees storage. So how likely is it that we would be able to store a solid dosage form at -110 degrees Celsius? It's simply not likely at all. And so it was remarkable that this team observed that menthol was able to be stabilized in the amorphous form at room temperature for 12 months. So mesoporous silica was successful in stabilizing this extremely poor glass former. And this clearly would not be possible with hot melt extrusion or spray dried dispersion. But the researchers here went one layer deeper. They wanted to probe on a molecular level what exactly is taking place within those pores that provide such unparalleled stability for this extremely poor glass former. And to do this, the researchers used a somewhat underused tool in formulation development, which is solid-state dielectric spectroscopy. Solid-state dielectric spectroscopy looks at the electrochemical response of a molecule, or a formulation in this case, and considers how the molecules are moving in that system. So it can be used to look at molecular mobility. And what the researchers found in this study is that when you confine menthol in that 6 nanometer pore environment, you have a substantial reduction in alpha relaxation. Alpha relaxation is one of the critical molecular mobilities along the path to recrystallization. So this molecule is totally hindered in space. It can't move around much. It can't recrystallize. They also observed a substantial, almost complete reduction in beta relaxation, Johari-Goldstein relaxation, sometimes referred to. So both alpha and beta relaxation, alpha and beta mobility, substantially hindered inside that 6 nanometer pore. Now, of course, no one is trying to develop an amorphous menthol formulation for the market. And so we wanted to repeat this study with a more pharmaceutically relevant compound. In this case, we turned to glibenclamide, and we successfully loaded glibenclamide onto mesoporous silica in the amorphous form. You can see the crystalline structure of glibenclamide there in black, the characteristic Bragg peaks on the XRPD. And in red, we have glibenclamide loaded silica. We no longer have those Bragg peaks. We have an amorphous halo because our molecule is successfully stabilized. So what's happening to glibenclamide within this 6 nanometer pore is identical to what happened to menthol. I'm not going to spend too much time digging into what these graphs show, so I'm just going to tell you the main conclusions. On the left-hand side, we have neat amorphous glibenclamide. That means glibenclamide without any kind of stabilization. And what you can see with the black line here is a very, very prominent alpha relaxation event, and also a very prominent beta relaxation event. However, when we take glibenclamide and load it onto mesoporous silica, our alpha relaxation is substantially reduced, and again, our beta relaxation is almost non-existent. So when we load pharmaceutically relevant compounds onto mesoporous silica, we substantially reduce their molecular mobility. And this reduction in molecular mobility opens up the door to stabilize pore glass formers in pharmaceutical amorphous formulations. So that's what we set out to show. We selected 2 extremely poor glass formers, carbamazepine and haloperidol, and we formulated both of these poor glass formers with hot melt extrusion and with mesoporous silica. We went through an entire optimization process for hot melt extrusion. We didn't just pick any old polymer off the shelf. We combined experimental with computational prediction to make sure that we had the most optimal polymer. We then loaded these molecules onto mesoporous silica, and then we characterized the formulations with XRPD and with non-sync passive dissolution. We then stored these formulations under ICH Q1A accelerated stability conditions of 40 degrees Celsius and 75% relative humidity. Then we assessed any changes in these parameters over the course of a 3-month stability study. Straight away off the bat, we observed that mesoporous silica was able to consistently stabilize high drug loads, whereas HME was unable to do so. Carbamazepine HME was crystalline in 30% drug loads. We were only able to stabilize 20% drug loads in a carbamazepine hot melt extrusion. However, for carbamazepine mesoporous silica formulations, we were successful at 30%. For haloperidol, the situation was even more dire, shall we say, we were only able to stabilize 7.5% drug load in the amorphous form in hot melt extrusion, whereas mesoporous silica was successful in stabilizing 30% drug loads of haloperidol. Now, as it turns out, haloperidol has a very low dose, so around 5 milligrams to 10 milligrams. So a 7.5% drug load is still a feasible formulation. But we all know that the average drug load is around 200 to 400 milligrams. So imagine how many tablets that patient would have to take if we could only stabilize 7.5% of that molecule in our hot melt extrusion formulation. But the comparison didn't stop there. We then stored these successful formulations under the accelerated conditions. So we took the 7.5% samples for haloperidol and the 20% samples for carbamazepine, and we stored them at 40 degrees Celsius and 75% relative humidity. Straight away, we could see macroscopic changes in our HME extrudates after only 1 week. On a previous slide, we discussed the concept of a solid solution. One of the biggest indicators of a solid solution is that your polymer becomes transparent. So you can see through the polymer because the drug is literally in solution inside that polymer. And that's exactly what we observed with our fresh sample. Our extrudates were transparent, we could see through them. We were pretty confident that we had a homogeneous distribution of our molecule inside the polymer. However, after only 7 days under these accelerated conditions, we had no longer a transparent extrudate, but instead an opaque extrudate. We can no longer see through our polymer, we no longer have a solid solution, and this is highly indicative that we have that phase separation behavior that I previously introduced. And that's exactly what we observed in our scanning electron microscopy images. Here we have haloperidol. Haloperidol loaded silica is on the left and haloperidol HME is on the right. Our fresh sample is on the top and our 1 week sample is on the bottom. No change in the silica was expected, no change was observed. However, for the extrudate, we've gone from a relatively nice homogeneous surface to a surface that looks totally different. It looks heterogeneous. We have these kind of globule-like structures on the surface. This really confirms that we have phase separation in our extrudates. Same was true for carbamazepine. We had no change from the fresh to the 1 week sample for silica, but for the HME formulation, we went from a totally smooth homogeneous surface to a non-smooth heterogeneous surface. And if you can see that, it's starting to look like crystalline structures on the surface of that polymer. So we no longer have the immobilization within a polymeric matrix. Our molecules are forming API-rich domains via phase separation. And as we discussed, we've just given the molecule the opportunity to recrystallize. These molecules are extremely poor glass formers. They are not comfortable in the amorphous form. So if you give them an opportunity to recrystallize, they will recrystallize. And that's exactly what we observed throughout the study. Here we have XRPD data. On the left-hand side, we have haloperidol-loaded silica, and on the right-hand side, we have haloperidol HME. The bottom XRD curve is crystalline haloperidol. Then as we go up the figure, we have the fresh sample, the 1 month sample, the 2 month sample, and the 3 month sample. Take-home message for haloperidol-loaded silica, we saw no recrystallization throughout the duration of the study. Haloperidol, which is an extremely poor glass former, was successfully stabilized throughout the study. And furthermore, we've continued on these measurements naturally, and we can now see 12-month and soon 18-month stability. These molecules are stable in the amorphous form with our mesoporous silica. The situation was slightly different for haloperidol HME. Again, crystalline haloperidol is at the bottom, then we have the fresh sample. And what you can see from the fresh sample is we are already seeing some evidence of microcrystallinity here. So it's not a perfectly amorphous structure. And these peaks simply grow as the time progresses. So the haloperidol is not stable in the amorphous form in this hot melt extrusion formulation. The same situation was observed with carbamazepine. On the left-hand side, we have carbamazepine-loaded silica. And again, crystalline carbamazepine on the bottom. And as we go up, fresh 1 month, 2 month, 3 month, same story as haloperidol. our carbamazepine at a 20% drug load remained amorphous for the duration of our study, and indeed remains amorphous to this day. For carbamazepine-loaded silica, we get the same scenario as for haloperidol. Carbamazepine simply is not stable in this hot melt extrusion formulation, and so over time in these accelerated conditions, we get recrystallization of our formulation. So both hot melt extrusion formulations, carbamazepine and haloperidol, would have failed the regulatory requirements for stability. And there's a good reason why these requirements are in place. Because as I already mentioned, if you have an amorphous formulation that recrystallizes while it's on the shelf and is then somehow given to a patient, that patient receives a totally different dose as someone who receives a sample fresh off the shelf with an amorphous formulation. Now, we used passive dissolution to show this effect in our study. In the interest of time today, I've not included the passive dissolution data, but if you are interested, I've included the reference to our paper at the bottom, and we have much more data digging into these systems and really looking at what was the impact of this change in solid state on the overall formulation performance. But ultimately, what we saw is that for the hot melt extrusion formulations, our dissolution profile was totally different throughout the study, and at the end of the study, there was no advantage at all. Whereas for the mesoporous silica formulation, we retained a strong, consistent dissolution profile for the duration of our study. So as already hinted to, this work was published in Pharmaceutics a couple of years ago now. Please go and check it out. It's a very interesting read. There's lots more data that I've been unable to share today. And in addition, my colleague Finn and I wrote a commentary on this topic in the American Pharmaceutical Review. And in that commentary, we really dig down into some of the literature on this glass forming topic, as well as the ability of porous silica to stabilize these type of molecules. So I hope that was interesting for you, and we do have some time for questions, which is great. And I hope you have lots of questions. But before we come on to the Q&A, let me quickly summarize what we've discussed today. Number one, amorphous stability is a key issue for the development of poorly soluble drug formulations. And glass forming ability is a critical physicochemical parameter that can ultimately predict what the long-term amorphous stability will be. Poor glass former molecules are unstable in the amorphous form, and as a result are difficult to formulate with traditional amorphous technologies. This results in a disproportionate amount of good glass formers on the market, and ultimately these poor glass formers would fail in development. With our data and with some previous data in the literature, we believe mesoporous silica is a great candidate to rescue these molecules by successfully stabilizing the poor glass former in the amorphous form. So thank you very much for your attention, and thank you to the organizers for giving me this opportunity to share this data with you. I hope it was interesting for you, and I hope it's maybe inspired some interesting questions, and with that, I would hand it over for the Q&A. So thanks, Shiva. I'll hand it over to you.
Thank you, Dan. This was a wonderful presentation, and as you rightly mentioned, we have a lot of questions coming in, so let's move on to the Q&A session. So the first question is, how much use does microprecipitated bulk powders get? Are they easier and faster in early phase development?
So I'm not super sure what that's referring to. Do you mean microprecipitated silica? So there is some different types of silica that is not porous in a way. I would say that's not used as often. If that question was referring to another topic, I'm not fully sure what the answer would be. I apologize for that.
Okay, I let the participant to rephrase the question later. Let's see if that comes across. So, okay, the next question is, what kind of unique characterizations are required for mesoporous silica-based amorphous formulation?
So I think the key parameters, the two key parameters, and I alluded to this, is the surface area of the silica and the size of the pore, because it's from both of those parameters that the functionality arises. So you need a sufficiently high surface area, otherwise you won't be able to absorb the molecule, and you need a sufficiently small pore size so that you get this stabilization and the reduction of the the reduction of the molecular mobility.
Mm-hmm. The next question is related to slide 10, 1-0. What is the maximum drug loading possible in mesoporous silica from your experience?
Yeah, so great question. I guess we don't need slide 10, but here it is. Yeah, so 30% is kind of a gold standard in amorphous formulation. So mesoporous silica does not struggle at these type of drug loads. What we have seen is that there is even some scope to go higher. So for example, celecoxib, which I've looked at in detail, you can even go as high as 50% drug loads and still stabilize the amorphous form. So I would say as a high chance of success, you can start with 30%. For most APIs, this works. And you may even have the potential to go higher than this, up to 50%. I've yet to encounter a molecule that is successful above 50%. Usually they just crash out in the crystalline form with the silica on the surface.
Okay. The next question is kind of putting you in a spot here. So the question is, I'm looking for a surfactant or some other material to stabilize the nanoparticles of size 120 to 150 nanometer. Could you please let me know how can I use the technique you mentioned here? Is there any possibility to use nanoparticles of different sizes ranging from 120 to 1000 nanometer? And is it easy to disintegrate the particles in solution without affecting the particle size?
Yeah, so I don't think silica is an option here, but fortunately, I think I can answer the question. Another part of my educational background is I developed a new method for determining optimal drug polymer concentrations using computational chemistry. And so something like that could be a really interesting tool to determine what optimal polymers or surfactants are to maintain those nanoparticles in solution. I know at least one of my colleagues did something similar with SNEDs formulations. They took the computational workflow that we developed and they applied it to SNEDs to find the optimal surfactant for that formulation. I'd be happy to provide that paper to you as well, Shiva. It's not really related to this topic, but I hope that at least was helpful and answered your question.
Sure, sounds good. The next question is, what is the difference between glass solution and solid solution at DSC curve?
So can you repeat that again, please, Shiva?
What is the difference between glass solution and solid solution at DSC curve?
So if you have a glass solution and a solid solution, would you see any difference in the DSC curve? Is that the question? Okay, so I think this is one that I would prefer to follow up with offline. I can find out the answer, but I don't want to waffle and try to make something up on the spot. So I'll follow up with that one offline if that's okay. Thanks.
Sure, sure. The next question is, will the GFA method work for a polymeric API that melts and recrystallizes as another polymorph in the first DSC heating cycle?
Yeah, so if it's a polymeric type molecule that recrystallizes in the first heating cycle to a different polymorph, then I guess the same principle holds. The GFA classification method doesn't differentiate based on what type of polymorph is recrystallizing. It only wants to determine if it is recrystallizing. So if you have a molecule with multiple polymorphs that change in that cycle, I think the key observation for that molecule will be, does it recrystallize? And if that molecule doesn't recrystallize, then you can quite comfortably say it's GFA class 3. If it recrystallizes on the second melt cycle, you can quite comfortably say it's GFA class 2. And if it recrystallizes straight away on melting, then I think you can say it's a poor glass former. I think the polymorph would not affect the overall behavior of the glass former.
Okay. The next question is, can you model or extrapolate shelf life estimates from dielectric relaxation data?
It would be great. I don't know if anybody's looked into that. I think this is an active area. So like I said, the dielectric spectroscopy is relatively underused in formulation. So this was one of the first studies with menthol that used it. We then collaborated with the New University of Lisbon, and those guys are really experts on that technology. And as of last update, I know they are starting to look at polymeric formulations with this technology. but I think it's still a relatively immature area of the literature. In principle, I think it sounds like a great idea, and it would be certainly very helpful also from a regulatory point of view if you can use the dielectric mobility of a molecule as a sort of, let's say, fail-safe for the stability. I think right now that's not been developed though.
Okay. The next question is related to slide 22. How did you measure the loading content of carbamazepine in mesoporous silica?
Yeah, so this is a good question. It's relatively simple how you determine the loading content. There are two methods. The relatively simple is that you take a known mass of your powder, let's say, for example, 1 gram. You can then disperse that powder in an organic solvent. This triggers the full release of the molecule from the silica, and you can then filter and centrifuge that sample and then take the eluents into the HPLC and determine the concentration of the molecule that was in that sample. And then you can calculate back. So I had this amount in my 1 milliliter of, let's say, DMSO. I had 1 gram of total bulk material. This corresponds to a 30% drug load. So that's the simple way. The more complicated way is using NMR. I don't know exactly how that process works because my colleagues in our NMR lab usually do that for us. But that's also, let's say, a more robust way of determining the load and concentration.
Okay. Can you go to slide 24 or 25? The question is, what polymer was used for HME experiments?
Yeah, absolutely. So I said we optimized the polymer. So we used both computational approaches, we used solubility parameter predictions, and we did experimental small-scale screenings. We looked at approximately 10 different polymers, and the best polymer for both of these APIs was polyphenol alcohol. That's what we used.
Okay. The next question is, for the HPNCAS and amorphous drug-loaded silica, are they just mixed together?
Yeah, so great question. In this case, yes. So we simply take the drug-loaded silica, we combine it with the HPMCAS in a simple blend, and this can then be tableted or put in a suspension or whatever. That works pretty well. One of the other studies that we worked on is we developed a different method. So instead of simply blending the polymer with the silica, we actually included the polymer in the loading solvent. And as a result, we get so-called co-loaded mesoporous silica with our polymer. And again, we published that work and it has some certain advantages, it has some certain disadvantages. And I can send that to you, Shiva, and you can distribute that to anybody who's interested.
Sure, that'll be helpful. The next question is, will mesoporous silica be suitable for tableting process?
Yeah, absolutely. So we weren't able to talk about that today, but our silica works incredibly well in a tableting process. Of course, we are just talking about silica, so it's not the best tableting ingredient, but when you combine it with your standard fillers and binders, you get a pretty passable and good tablet. And for anybody who's interested in that, I can send some more information on galenical properties, flowability, compressibility, tablet hardness, etc. But take-home message is that, yes, absolutely, it's compatible with standard tablet processes.
Okay. The next question is, are there any approved drugs in the market using this new formulation technique?
Not yet, but there will be. So we're very close to one of our customers in Asia having a formulation on the market. So that's very close, either the end of this year or the beginning of next year. We also have a very rich pipeline of customers using this technology in phase 1 to phase 2 clinical trials. But from a rate, maybe if I can preempt another question since the person can't respond. From a regulatory perspective, there is very little concern. So as I mentioned in the introduction, silicon dioxide has been used as a glidant for decades. And so it's multi-compendial, so both the United States and the European Pharmacopoeia, and it is also generally regarded as safe by the FDA because we're talking about silica with micrometer particle size. We are not talking about nanoparticle silica. nanoparticle silica can be absorbed from the gastrointestinal tract, and so therefore there's some tox concerns, but we're talking about micrometer particles. These particles cannot be absorbed by the gastrointestinal tract. They simply pass out in the waste, and as a result, they're generally regarded as safe GRAS by the FDA.
Okay. The next question is, what are the disadvantages or limitations of using mesoporous silica for amorphous formulation?
Yeah, there are two main disadvantages that are similar to the standard polymeric technology. So the first one is drug loads. I already mentioned that, in some cases we can get a 50% drug load, but 30–40% is the standard. That can become a limiting factor if you have a very high dose per day. There's only so much you can get inside the tablet and still have a reasonable tablet size. But this is a well-known limitation with amorphous technologies in general, and it's simply something that we can't do anything about. The other limitation, and this is one that is shared with the spray dry dispersion process, is that you need to solubilize your API in an organic solvent. So if you have a real brick dust molecule that doesn't even go into solution in an organic solvent, then it's very challenging to use mesoporous silica as a technology, and that is also the case with spray dry dispersion technology. So two main points, dosage and compatibility with solvents.
Excellent. The next question is time for two more questions. So does the preparation process require a special equipment, especially in the preclinical small scale?
Yeah, I'm so glad this question's asked. Thank you for that. No, absolutely not. So one of the biggest selling points for mesoporous silica as a technology is that at a very basic level, all you need is a rotary evaporator. So you don't need an extrusion equipment, you don't need a spray dryer. If you've got a rotary evaporator in your preclinical lab, you can load molecules onto silica. And one more advantage from a preclinical perspective is that you can suspend your mesoporous silica formulations in a suspension, like a methicel suspension, and you don't get premature drug release. So sometimes if you have a spray dry dispersion and you suspend that SDD in a methicel suspension, then you already trigger drug release, dissolution, and precipitation. Whereas when you suspend mesoporous silica in a methicel suspension, the methicel suspension cannot absorb because it's too viscous, and so therefore the drug is left inside the silica.
Okay. This is the last question for the session today. Can the loading be conducted by other methods in addition to rapid mixer granulator? And what is the maximum loading capacity? I think you already answered that. So.
Yeah, so loading capacity we've covered, up to 50 is possible, 30 to 40 is average. Yes, other methods can be used, and by all means try. You can do melting-based processes onto silica. Mesoporous silica has an incredibly high melting point in the thousands of degrees Celsius, so that's not a concern. It can be quite challenging to fully mix a melted API with silica. You also run into thermal instability issues. Other people have used supercritical fluids. That's really interesting. It's not something that I know a great deal about, but it's certainly possible according to the literature. But by far the most predominant method is a solvent-mediated loading process.
Great. That pretty much ends our live Q&A session today. Thank you so much, Daniel, for the wonderful presentation and for detailed answers to most of the questions today. So let me...
Yeah, thank you. And thank you everyone for your attention.
Okay. Cool. Thanks, Daniel. Really appreciate your contribution to the FSF 2021 webinar series. So before we end the session today, I'd just like to give you a heads up on what's coming up next for webinar number 5. So pretty much at the end of April, finished 4 webinars. So we're moving into May. We're starting with Dr. Luca Russo from GSK. Luca is a subject matter expert in crystallography, and he overlooks late-stage development assets within the material science group at GSK in Stevenage. Luca manages the single crystal XRD facility and provides structural insight into physical properties of crystalline forms in development. So next week, Luca will present cases where crystallography has been used in combination with molecular modeling to address challenges in structure determination, as well as in understanding solid form properties and risk management. The title of his talk is current practice and future perspective in crystallography for API solid form risk management. So don't forget to tune in next week on May 5th at 11 a.m. EDT. And as I mentioned before, there's still a lot of seats available. We have increased the seating capacity for this FSF 2021 webinar series to 1,000 from 500 last year. So right now we are at the 750 mark. So we still have 200 seats available. So please continue to spread the word with your friends and colleagues in the industry and encourage them to register for this webinar series. The registration is absolutely free. As I mentioned before many times, this is all about you, not about the organizers. So please tune in next week, and I'm looking forward to seeing you all again next week. And if you have any feedback, comments, or questions about the webinar series, and also anybody is willing to give a presentation as a speaker, willing to contribute to the series, please reach out to me with the topic of interest, and we'll get in touch with you. Okay, so see you next week. Until then, take care, stay safe, and thank you very much for your attention today. Bye.